guys. My name is Mark, and I'm going to talk to you today about a project in which I tried to figure out a method to generate comprehensive venom information, venom sequence data from any cone snail species, and then what we can learn about venom evolution with that comprehensive venom data. So the goal of my research program is to try to understand the genetic basis for adaptation and diversification, and I do this by studying venomous animal systems. So the use of venom for predation has evolved several times in organisms such as snakes, snails, and spiders. And venomous animal systems present an excellent opportunity to study the genetic basis of adaptation and speciation because of a relatively simple link between genotype, phenotype, and ecology. So venoms are typically composed of complex mixtures of toxic proteins and peptides that are encoded by multi-locus gene families that undergo positive selection and gene duplication. These venom proteins have a direct impact on a species' ability to capture and apprehend prey. And so you have this nice relationship between genotype, phenotype, and ecology. The organisms I study are the cone snails because they are the least terrifying of all the venomous organisms. The cone snails represent a hyper-diverse group of carnivorous marine snails that comprise of over 800 species. And they typically feed on either worms, mollusks, or fish. And they capture their prey by injecting a cocktail of neurotoxic peptides that's synthesized along the venom duct and is injected into their prey with a radula that's modified to be like a harpoon or spear. And so radula are analogous to teeth in snails. One of the challenges of working with venomous animal systems is that it's generally very difficult to generate venom sequence data. So much of what we know about patterns of venom evolution comes from a restricted set of species from a restricted set of gene families. And this is in part because of the methods by which we generate venom sequence data. So the main way in which we've done this in the past, not me specifically, but other people, have been through cDNA cloning. And so sometimes a study will have to sequence hundreds of clones before they're pretty sure that they got those 20 sequences from that one gene family, from that one species. RNA-seq and transcriptome sequencing have greatly expanded our capacity to discover venom genes and venom transcripts, but this represents an incomplete picture of venom diversity, where some studies have shown that in cone snails, at any one given time, an adult individual will only express a fraction of the genes available in their genome. And so I tried to figure out to improve upon these previous methods by trying to figure out a target sequencing approach to generate comprehensive venom information. So to do this, I had to first identify the targets that I was after, and so I sequenced 12 venom duct transcriptomes from species spanning the phylogenetic breadth of cone snails. And the goal for these transcriptomes was to identify both markers that could be good for phylogenetic inference and also to target the venom genes. And so you've probably heard a whole bunch of talks about phylogenomics. It's like pretty straightforward to use exon capture or like UCEs and capture approaches to generate large-scale phylogenies. The problem with venom genes is really intense. So this is a single venom gene alignment from a single species from a single gene family. And like how I feel about my life right now is a big mess and a little bit incomprehensible. And so if I only knew one of these sequences and I put that in my bait or target design, it is very likely that I wouldn't be able to recover anything else from this species in this one gene family, and there are dozens of gene families. And so what I ultimately did was, on this bait design, I have about 5,000 exons from common cellular genes that I thought was good for phylogenetic inference. And what I did for the venom genes was I targeted the entire coding region plus 100 base pairs of the untranslated regions on either side, because it's been shown in previous studies that these untranslated regions happen to be, for whatever reason, conserved between species. And it was my hope that these interesting coding regions would stay attached in the genome to these UTRs and not split up into really tiny bits of exons. Okay, and so what I did was I performed a preliminary pilot study. I sequenced 32 species and performed this hybridization experiment, 12 of which came from the species for which I had transcriptome sequencing, and 20 of which I just had and threw on and hoped for the best. And first thing is that this is the phylogeny from those 5,000 phylogenetic markers, exons. It's a racemel tree with bootstrap with nodes. It's highly supported, and if you care about the diet, they're colored. And I got about 95x coverage, and so this is pretty straightforward thing to do. For the venom exons, when I did recover them, they were also at high coverage, so about 150x. And of the 49 gene families that I targeted, about 34 of them had high capture success, which I defined as the transcript I targeted being tiled with exons across 80% of the transcript and at least 80% of the genes in a gene family that I targeted. And this, in total, generated over 10,000 unique toxin sequences, which is around 2 to 4 times the number of toxin sequences that have ever been generated over several decades of work. So that's really cool and crazy. Okay, and so with all this comprehensive venom information, we can learn a little bit about their genetic architecture and gain some new insights into known molecular evolutionary patterns in venom. And so the first thing I'm going to tell you about is what a typical venom transcript looks like. And so you have the UTR regions on either side, and this is the coding region. And two interesting functional regions that I want to point out to you is first the signal region. And so the signal region consists of about 20 hydrophobic amino acids that tells the cell, like, hey, secrete this protein. And it's generally conserved across between species, and it's what's used to categorize these transcripts or these genes into different gene families. And then this is in contrast to the hypervariable mature region, which ultimately becomes the mature peptide. And that's the part that was super incomprehensible in that alignment I showed you earlier. And this whole region right here is about 100 to 200 base pairs long. And so there's just this extreme rate variation across a short stretch of DNA. And so a hypothesis for why this extreme rate variation is allowed is that there exists on different exons in the genome. And so that's the hypothesis that's put out in literature, and now we can test it because I got these venom information, these venom sequences from genomic DNA. And so this is the largest proportion of these regions on a single exon in the genomic data that I generated, and in most cases, the signal region and the mature toxin region are combined to single exons and are often separate from each other. And so what I wanted to figure out now with this genomic data is how the adjacent regions look and what their molecular evolutionary patterns look like. And so what did I do? Okay, so I looked at pairwise comparisons for each gene family. I looked at all possible pairwise comparisons, looking at pairwise differences between the coding region and the immediately adjacent non-coding region. So this could be the UTRs or the introns, and to compare molecular evolutionary rates of divergence. And so for the signal region shown here in blue, they follow the general common pattern that we think for most common cellular genes, where the coding region, the exon, is much less diverged relative to the non-coding regions. And so they're about 20,000 points here. The exact opposite pattern is seen from the mature toxin coding region, where the exons are oftentimes 2 to 3 times more divergent than the immediately adjacent non-coding regions. And so there's a couple of points from this pattern that I can make, is that first, this is a clear case of positive selection, and it is also seen in snake venom proteins. You also see this pattern in the genome, and also in sperm fertilization proteins in abalone, so other systems for which genes are under positive selection. And the other interesting thing about this pattern is that if you have a system in which you have rapidly evolving genes, you can use targeted sequencing methods to pick up a whole bunch of those genes from a whole bunch of different species by targeting these non-coding regions, because they tend to be relatively conserved. And this seems like a general phenomenon for genes under rapid diversifying selection. Okay, so another thing that happens in venom evolution is gene duplication, and so now we have comprehensive venom information for that. So this is a time-calibrated phylogeny, and here I have some information with numbers that you can clearly see from the back. This is total conotoxin diversity, which is the number of genes that's found in the genome of each of these species. And so I'm going to zoom in on a couple. We can see, with comprehensive venom information, we can sort of see the extent of this variation now. And so, for example, it goes as high as 859 venom gene copies in Conus coronatus, to as low as 120 in Conus papilliferus. And so, I guess 850 is sort of pretty high for a trait like this. The closest genome is the owl limpet, and it has about 24,000 protein coding genes. And so 800 out of 24,000 is 3% of the coding regions are devoted to venom. So that's pretty cool. And what I have here, with these values, I reconstructed sort of the reconstructed the evolutionary history of expansions and contractions using a program called CAFE. And their color here on this phylogeny, where green is a net expansion and black is a net contraction. And basically the color scheme shows is that gene gains and loss are prevalent and present throughout the history of this group, and is a big contributor in determining like the genomic availability of venom genes that these species can use to capture their prey. And when you zoom in on any of these individual gene families, so I have 3 of them here, one interesting thing that I found was that there was clade-wide patterns of expansions and contractions. So for example, this clade here of Conotoxin, Aristophanes, and Malares expand in these 3 families and duplicate a whole bunch of copies, and their immediate sister clade doesn't diversify to the same extent. And in some cases, entire gene families go extinct across clades. And I saw this pattern across 50% of the gene families, over 50% of the gene families that I looked at. And so I'm still having a little bit of difficulty explaining what this means, but I think it suggests that history plays some role in determining the number of genomic elements that they have for venom protein production. And then finally, in one previous study, it's shown that Cone snails will only express 40% of the genes in their genome at any one given time, but that was only inferred from one gene family. And this is averaged over several dozen gene families, and we have several species here to sort of see the extent of variation. And so it goes as high as 63% in Conus californicus, expressing 63% of its genome, expressing 63% of the available venom genes in its genome, compared to Conus ebrius, where it only expresses about 23%. And there are suggestions in the literature about why this might, that's very scary, about why this might be. Like, cone snails have some cone snails have large geographic distributions, and in these large geographic ranges, they encounter different prey in different spots in their range, and also have to compete with different congeners. And that likely has an impact on when genes are turned on, and often likely influences these patterns. And so finally, where I'm going with this, and why I tried to figure out this method, was that I wanted to generate comprehensive venom information across most of the diversity of this group, so that we could understand how the evolution of these genomic elements was impacting patterns such as speciation rates and rates of morphological evolution related to the venom apparatus. And so I just, like Thursday, finished library preps and these captures for 360 species. It took 5 years to get to this point. So hopefully the data is great and has the potential to let us understand the genetic underpinnings of speciation rates and stuff like that. And with that, thank you.
So thinking about having more copies of these venom genes, do you know if that's related to venom toxicity or perhaps something else?
The only thing I can say about what the number of copies means is that I found in my work that dietary breadth is related to the number of copies in a species genome. And so if you eat more things, you have more genes in your genome, and this relationship is much stronger when it's only restricted to express data. Yeah.
Are all these copies functional?
I did not look for evidence of pseudogenization in my study because I couldn't figure out a way to parse that out in my data. These proteins are very short, oftentimes 10 to 33 amino acids long, and if I find a stop codon somewhere, I don't know what's the difference between a 10 amino acid versus a 13 amino acid protein. So